Oogenesis. It is the process of formation of the functional haploid ova from the diploid germinal cells in the ovary or female gonad. Early stages of oogenesis occur by the time female fetus is only 25 weeks old. At this time, all the oogonia have been formed and entered into the primary oocyte stage. Multiplication phase. 10 cells of the germinal epithelium ovary, which are larger than the other functional germ cells, they undergo repeated mitotic division to form diploid oogonia. Some 2 million oogonia gamete mother cells are formed in each fertile ovary. The oogonia form egg tube of P. plugger which grow into the cortex and give rise to a multicellular mass called Oogenesis. Mm, Growth phase. One oogonium of the egg nest grows in size and function as primary oocyte. The other cells of egg nest form an epithelial covering over it. The same is called follicular set or epithelium. The in-seated primary oocyte is called primary follicles. A number of these follicles originate between the birth and puberty. At puberty, an ovary contains 60,000 to 80,000 primary follicles. Maturation it begins in the pubertal stage of females, but it is interrupted quite early. The primary oocyte begins meiosis 1, but the division is arrested in the reproductive stage of prophase 1. It is in this phase that ovarian follicles containing the oocyte occur in the coitus and remain so till the female attains puberty. After attainment of puberty, follicles start and their content resume development, usually one at a time and once a month. Follicles differentiate into layer of granulosas or another layer called theca develops from the cortex. With the formation of the theca, primary follicle transform into the secondary follicle. Theca differentiate into two layers, theca externa and theca interna. Granulosa cells secrete fluid that cause development of the cavity or antrum around the primary oocyte. The stage is tertiary follicle. Primary oocyte grows further and meiosis 1 it produces a large secondary oocyte and a small polar body. Both are haploid. The polar body has a very small amount of cytoplasm, but the whole chromosome set. In human females, the polar body does not divide further. The bulk of nutrient-rich cytoplasm is retained in the secondary oocyte. The follicle grows to maximum size and is called Graafian follicle. Zona pellucida develop around secondary oocyte. The secondary oocyte proceeds with meiosis 2, but the division gets arrested in metaphase 2 stage due to accumulation of metaphase promoting factor in this stage of oocyte that ovum is set during ovulation. It passes into oviduct where in the ampulla part cell cycle will resume only after the entry of sperm. It triggers the breakdown of APF and promotes synthesis of anaphase promoting complex meiosis 2 is completed. A second polar body is extruded. The oocyte is now changed into ovum or oocyte, which soon gets changed into zygote by the fusion of male and female pronuclei. Hormonal control of oogenesis. In response to production of GnRH or gonadotropin releasing hormone, anterior pituitary secrete 2 hormone FSH follicle stimulating hormone LH luteinizing FSH stimulates follicular growth and maturation of oocyte granulosa cell of developing ovarian follicle produce estrogen in the presence of high titer of both estrogen and ovulation occur. High concentration of estrogen inhibits secretion of estrogen and GnRH. This negative feedback control let's help in convert rupture Graafian follicle in corpus luteum. The later secret progesterone separates the uterus to receive fertilized ovum. High concentration of progesterone inhibits further release of LH from the anterior pituitary GnRH from hypothalamus. This is another example of negative feedback loop. Ovum. It is non-motile female gamete. Human ovum is rounded, non-cleidoic without cell and allocetal without yolk. It is about 100 in diameter. Ovum is covered by its own membrane called plasma lemma or ovalemma. Its cytoplasm is ooplasm. In the outer region, ooplasm contains a number of mucopolysaccharide cortical granules. Cytoplasm contains microtubules, mitochondria, ribosome, RNA, protein, glycogen particle, and lipid globules. Nucleus is large. It is also called germinal vesicle nucleus. Generally in metaphase 2 stage prior to fertilization. Typical nucleus or pronucleus appear after entry of sperm. Egg or ovum has a polarity. There is an animal pole and vegetal pole. One or two small pole bodies occur inside the outside the plasma membrane near the area of animal pole. The space where the polar body occur is known as perivitelline space. Ovum has two covering a non-cellular genera pellucida and multicellular corona radiata. Genera pellucida is a thick layer of protein-rich covering which is secreted by joint activity of secondary oocyte and inner follicular set. It carries receptor protein ZP1, ZP2, ZP3 for determining attachment or non-attachment of sperm. Genera pellucida also bears in growth of follicular cell for transport of nutrient and biochemicals to the egg. Corona radiata is part of granulosa cell which remains around the ovum at the time of ovulation. Its cells are radially elongated. They are held together by mucopolysaccharide called hyaluronic acid. Similarities between spermatogenesis and oogenesis. They are meant for producing male and female gametes from germinal cells of gonads. Both the process involve three phases: multiplication phase, growth phase, and maturation phase. Mitotic division occurs in multiplication phase. It increases the number of spermatogenia and oogonia.